Thank you, David. Good afternoon to all. My name is Emran Ali. I'm a faculty member, Department of Plant Pathology, University of Georgia, Tipton campus. Today, I'm going to talk on molecular detection, gene expression, and phylogenetic analysis of Aspergillus flavus strain isolated from peanut seed lots in Georgia. Definitely, I should first acknowledge this work done in collaboration with Dr. Calvert and Dr. Brenneman with my students. Without their help, this work is not possible to conduct. At first, I'd like to give an outline of my talk. So I'd like to mention a brief overview of Aspergillus flavus problem in peanut seed production in Georgia. Secondly, I would like to mention how we characterize the Aspergillus isolate we collected from Georgia in different seed lot. Thirdly, I would like to mention about their aflatoxin status in relation to the gene expression study. Finally, I'd like to give you a brief outline what's next we will do for this research. In this right-hand side photograph for our new PhD student, Mackenzie Khan, who mostly involved in this project and conducted many of these research. So what's the issue? I think the previous two talks, we are clear about that. In 2020, we have a huge issue regarding our peanut seed contamination by Aspergillus. The reason why the traditional seed treatment chemistry available on that time dynasty did not act properly, so that seed treatment's failure, and as expected, we have seen lower germination. So on that time, we focused our research seeking a couple of answers. Why the seed treatment did not work? That part, we have done research and we have already published. And the second part was to see whether or not these isolates are solely Aspergillus flavus. If indeed they are Aspergillus flavus, are they alpha-toxicogenic? If the alpha-toxicogenic, are all these isolates are same? Is there any genetic variation among these isolates? Any effect that cause more or less alpha-toxin production? All those questions are related to the second part of my research, which I am planning to present. Before going there, I'd like to just give you a brief outline what we did last year and already get published. We have done the QI fungicide, especially azoxystrobin. So why the dynasty failed? So we try to understand that reason. As you can see in this photograph, we have tested in vitro sensitivity analysis all 104 isolate, and we have seen almost highly resistant and moderately resistant, almost 70% of isolate that is still 100 ppm they're growing. So there definitely is a problem going on. We did molecular analysis, especially the beta-tubulin gene we sequence, and we do see there is two well-known mutation, F129L and G143A. These two mutations present among these isolate in higher percentage. So that makes sense to why this fungicide did not work properly to controlling Aspergillus flavus. This work has already published in International Journal of Molecular Science. Now I'd like to move on the second part of our research. So here we did characterization of this isolate. At first, we have determined their ID based on this phenotypic characterization. The ideal colony morphology, a greenish color. So we have narrowed down it is Aspergillus flavus. Further, we confirm this isolate based on molecular characterization using Aspergillus flavus specific primer. And we do see very specific fragment that can confirm further that those isolate indeed Aspergillus flavus. In our experiment, we have tested 104 isolate. We did not see any other isolate other than Aspergillus flavus. To see all those isolate are very similar or they have molecularly variable. So we have done phylogenetic analysis based on beta tubulin gene. First, we have analyzed the Aspergillus isolate we have from Georgia. We did not see much variation among them, almost 99 to 100 ID are very similar. However, when you compare this isolate in NCBI's gene bank, previously submitted some other birds, other Aspergillus flavus isolate, we do see they have some variation among Aspergillus isolate compared to our Georgia isolate, and definitely all other Aspergillus flavus has a significant variation. Then we move forward to see whether or not all our Georgia seed lot isolate are any relation to aflatoxin production. To go that far, we first detected the aflatoxin production biosynthesis 4 gene, 3 structural gene D, Q, and P, and 1 regulatory gene R. What's their status in their genome? As you can see in this bar graph, we have seen this regulatory R gene, almost 85% isolate possessed this gene, and all other structural genes somehow 30 to 20% isolate they contain. In terms of how many genes in one isolate we have categorized in this table, all four genes present, 5% isolate, then like 10% isolate, or 17%, 2, 3, 4, or just one gene present, or just 0 gene present, 10% isolate, just no gene. So the next experiment, okay, we understand the gene is present there. So how they express? So we use the real-time qPCR analysis, and we have seen our Georgian isolate compared to the non-aflatoxigenic isolate, aflR. Our isolate, the expression of this certain gene, D, Q, P, and R, are really variable. So they're highly expressed to very lower expression. So their range is so high spread. So our data suggests this isolate, we have indeed a great variation among their gene expression, in particular this 4 gene. Now the question comes, okay, we do see these 4 genes somewhat present in different isolate, and they indeed have some very expression higher to lower. Do they produce aflatoxin? To answer this question, the next experiment we did to determine the aflatoxin on this plate before giving any stress or any things. Just plainly, we try to understand what is this isolate is really aflatoxin-genic. To answer this question, we have modified slightly a protocol from getting a measurement directly from the plate using the ELISA assay. And we do see all of our isolate, almost 20% isolate, we do see they are aflatoxin-genic. They indeed produce aflatoxin, especially AFB₁. And 80% isolate, we do not, although they have, as you see, like 85% isolate has indeed the regulatory gene present. However, for this isolate, when you measure aflatoxin-genic or non-aflatoxin-genic, 80% isolate did not produce any aflatoxin. Here's the overall scenario. I'm not going to detail this small case. So basically, in our result, if we summarize, we have done RT-PCR to see these gene presence or absence. Then we have done RT-qPCR to see if they present, how much their expression. And we also see the amount of aflatoxin, especially the AFB₁, the amount, if they present, if they produce, and how much they produce. Here's the summary of this table. We have categorized, divided into 4 groups. The 5% isolate who have indeed the 4 gene present, then we have seen 100% are positive for aflatoxin production. The second group, the 3 gene present in different combination, we have seen 20% positive if it is AF1D plus regulatory pathogen AF1R. So the same combination, there's 3 combination, whoever has AFD and R, we do see positive. The other 3 combination without D, we did 100% negative. The C group, these 2 gene present, we have seen 40% positive, and without D, just 100% negative. And group D, the 26% isolate pauses in this group. So we do see only the regulatory gene present isolate 20% positive. Without regulatory gene, we do not see any aflatoxin production status. That brings us like a total scenario, like could be the AF1R and aflatoxin regulatory gene plus the structural gene D put together has some role for aflatoxin production. So from this research, we summarize the isolate we have received that was indeed Aspergillus flavus, and among this isolate, they must have some genetic variability. 4 important genes in this study we studied are assessed. They're mostly involved in biosynthesis pathway. Our study demonstrated that aflatoxigenic and transcriptional diversity among these strains. And as I mentioned earlier, the expression of regulatory gene R and structural gene D could be correlated with aflatoxin production. Then what's next? The next is we are thinking to investigate ATFC gene. There is a BG transcription factor recently, likely Jack Fountain published in 2020, where they claim this specific gene or factor have contributed the pathogenicity as well as oxidative stress tolerance that could reflect more aflatoxin production. To examine whether or not in our Georgia seed lot isolate, they also contain this specific transcription factor. So we will examine using the PCR approach. We also will investigate the oxidative state H28O2 to see after treating with H2O2, is there non-toxicogenic isolate can be able to produce again aflatoxin or not. And thirdly, we'll also analyze the expression after different treatment of H2O2, especially this ATFC gene, to see is there any relationship among this gene expression and in terms of aflatoxin production. In overall, we're also targeting to sequence whole genome for 100 isolate to get better understanding the pathogen biology and their aflatoxin production. Finally, we also want to continue our work to evaluate the sensitivity to different seed-treating fungicide. As I mentioned in my second slide, the azoxystrobin, we have already conducted the in vitro sensitivity analysis, but still we're working on some other chemistry, fluorodeoxynyl prothioconazole, fluopyram, and the PDFlumeton, the SDH, the new product, and thiobendazole. So we will continue to work on this to check their in vitro sensitivity level for controlling Aspergillus flavus isolate. With that, I'd like to thank you for my student, postdoc, lab tech, and collaborators, and finally the funding agencies to support their support for conducting this research. Thank you so much. That's all my presentation.
Thanks a lot, Emran, for the presentation, and thanks for being here today and providing some really good information. I think because of time, we'll move on.